Hello, this video is going to go over Chapter 18, Objective 5, which is going to classify and characterize white blood cells. So as you can see in the picture there, the white blood cell is the one that kind of looks kind of spiky and is kind of white. All those red ones, well, they're red blood cells. Okay, so another word for a white blood cell is a leukocyte. Leuko means white, cyte means cell. Now, the white blood cells are part of the body's defense system against disease. They protect the body against invading microorganisms, fungi, bacteria, parasites, and also body cells that have mutated DNA. These would be cancerous cells, potentially. Leukocytes also clean up debris. They originate in the same place as red blood cells do. So red blood cells, white blood cells, platelets, they all come from hematopoietic stem cells in bone marrow. Now, a hematopoietic stem cell is a cell in the bone marrow that has the ability to make a red blood cell, a white blood cell, or a platelet. So it could make any one of those three. So red blood cells come from there, as do white blood cells. Like we said previously, red blood cells, you've got the most, as far as cellular components of blood go, there's more red blood cells than anything. Next are the platelets, and the least numerous in the blood will be the white blood cells. Now, I'm not going to ask you the number here. I'm not going to ask you to tell me it's 5,000 to 10,000 per microliter of blood. I'm just giving you numbers so you can actually see numerically that the white blood cells are the least numerous. But again, I'm not going to ask you the specific number. They are larger than red blood cells. If we go back a slide, you can't really see it in this one because of the type of white blood cell it is, but these white blood cells can be huge in comparison. So this right here looks about the same size as a red blood cell, but again, that's a specific type of white blood cell that does that. Most white blood cells are huge in comparison. They have a much shorter lifespan than that of a red blood cell. White blood cells can last minutes to hours. So they don't really last a terribly long time in comparison, overall, white blood cells overall, in comparison to red blood cells. They can leave the blood vessel through what's a process known as diapedesis. So let's take a look at that a little bit more. What you're seeing in the image above, you have a capillary. Now think about it this way. You've got white blood cells traveling through the capillaries, traveling through the blood vessel, along with red blood cells and platelets. The problem is if you get some type of invading microorganism, it may not be in the blood system. It may be in the tissues. So how does the white blood cell get from the capillary to the tissues to go take care of the foreign invader? Well, it's got to get out of the capillary. Now, I know capillaries are porous, but they're not that porous, not enough for a white blood cell to just pass right on through whenever it wants. So it exits through what's called a process known as diapedesis. And if you look, this white blood cell in the picture, you can see it's a multi-lobed nucleus, which is represented in purple there. Look at what happens to it. It kind of flattens out. Then it's allowed to squeeze through the epithelial cells that make up that wall of the capillary. Then they can go out and attack that foreign invader. So they've got to get out of the blood vessel to be able to do that. So let's take a look at diapedesis. It's a really short little video clip here. I mean, if it's 30 seconds, I'll be amazed. Okay, it's 11 seconds. I told you all it was going to be super short. So what you're seeing here, see this white blood cell right here? You can see the grayed out nucleus there. You can see the red blood cells, and you can see this pink right here, which is that inner wall of that capillary. Well, as we progress down, what's happening is, notice where the red blood cells are. They're floating, and they're not really up against the wall, but that white blood cell has got like Velcro on it, and the capillary wall has the other side of Velcro. So it slows the white blood cell down so it knows where to exit the capillary at. That way you don't have like this mass exodus of white blood cells through all your capillaries. It's just going to exit where these little finger-like projections stick up on the capillary. It's only going to exit there. So the capillary is like, hey, stop here. So nowhere else does it have these little finger-like projections that are visible. Again, they only come up when the white blood cell has to exit in that location. And the white blood cell has the receptors on its membrane, so that way it kind of sticks to that capillary wall. And it's going to slow down. The red blood cells are going to pass it. And then did you see what happened right there? That white blood cell starts to flatten out and change shape so it can easily squeeze through the cells that make up that wall of that capillary. That's called diapedesis. Now that white blood cell is able to go out into the tissues and go take care of that foreign pathogen that has entered into the tissues. So there's leukocyte classification. You're definitely going to need to know this. So you've got the 5 kinds of white blood cells that we talked about, right? Never let monkeys eat bananas. So let me get my little pointer thing. So these right here, the neutrophils, the eosinophils, and the basophils, never and the eat bananas ones, okay? These are the ones that are granular. You can see inside that cytoplasm how grainy that cytoplasm looks. That's because it's a granular white blood cell. You can see here that purple odd-shaped nucleus. That's typical for a neutrophil. It's a segmented nucleus is what that's called. The eosinophil, you can see right here, the cytoplasm has red granules in it. Now, these are stained that way, okay? You're not going to look inside the blood without any stain and see the eosinophil having red granules. It's you have to see the white blood cells under a microscope by staining them. And when you get to the microbiology class, you'll learn about staining blood, a blood smear, and then that'll pick those red dye up. Basophils are the granules in a basophil are going to stain like a dark blue, dark purpley kind of color. And you can see again here that odd shaped nucleus that's typical for white blood cells. So you need to know that your neutrophil and your eosinophil and basophil are your granular white blood cells. And you can see then the other two must be agranular, meaning A means not. So you can see with these lymphocytes right here, you've got different kinds of lymphocytes. You've got natural killer cells. That sounds really cool, doesn't it? B cells and T cells. Again, these 3 right here. Now, you can't look at these 3 and when you're looking under a microscope and say, oh, that's a natural killer cell or, oh, that's a B cell. All you recognize them as under a microscope is lymphocytes. Okay, lymphocytes. And the reason why I said, oh, remember that one that we saw at the initial page there? And I said, oh, that white blood cell, it's the same size as a red blood cell. That's not typical. Most of them are huge in comparison. Look at the nucleus of these lymphocytes. They're kind of rounded, okay? Unlike the other ones, which are segmented and lobed. Look at the size of that nucleus. That's about the size of a red blood cell. So in that first slide on this objective, that was actually a lymphocyte that we were looking at because it was the same size as a red blood cell. If you look at the neutrophil, the eosinophil, the basophil, and the monocyte in comparison, they're huge. So the only ones that kind of have that same size as a red blood cell is a lymphocyte. And then monocytes. These are huge. Again, I know it says they're agranular and it kind of looks like there's grains in there, but again, that's not going to show up on a stain. You're really going to see those granules on a neutrophil, eosinophil, and basophil. So this is really going to have some agranular cytoplasm in it. Now, the life cycle of a white blood cell, right here, you can see the red blood cells. They're going to be those light pink donut type things. You can see the size. This is actually a real smear, by the way, of human blood. You can see some platelets right here. That's actually a platelet mass. That's actually a lot of platelets. Normally, they're just little red dots like this one right here. The white blood cells, you can see they have been stained. They're quite a bit larger. So here's your red blood cell, and then there's your white blood cell in comparison. Again, these are just typical neutrophils. You can see right here this lymphocyte. That's actually a pretty big lymphocyte, to be honest. And then this monocyte over here, you can see that odd-shaped nucleus there. It's not very round. And you can't really tell by the picture here, but that is agranular cytoplasm. But again, you're really looking at the size and the shape of that nucleus to really identify that being as a monocyte. But most white blood cells have fairly short lives, meaning they're not going to last very long, hours to days for the most part. They get produced in the bone marrow, just like red blood cells and platelets do. But if you remember back in chapter 17, what was the hormone that told those hematopoietic stem cells in the bone marrow to make red blood cells? Do you remember that one? It was made by the kidneys. That was erythropoietin, EPO. So when the kidneys secreted EPO, when there was when the kidneys detected low oxygen, they would secrete that hormone EPO. And that EPO would travel through the bloodstream because that's what hormones do. And when it arrives at the bone marrow, the hematopoietic stem cells say, oh my gosh, EPO is present. We need to start cranking out red blood cells. So that was the trigger to let the hematopoietic stem cell know what blood cell it was supposed to make. But if colony stimulating factors and interleukins are present, that lets those same hematopoietic stem cells, oh, I'm supposed to make white blood cells now. So again, you're not going to get, if you're going to make something different, you have to give it a different signal. So for a red blood cell to be made, that signal is EPO. For a white blood cell to be made, that signal is going to be colony stimulating factors and interleukins. So I'm going to go ahead and give you guys a warning on this next test, chapter 17 and 18. I know that from the nervous system unit that we previously did, CSF stood for cerebral spinal fluid. If you see CSF in this chapter, 17 and 18, it's referring to colony stimulating factors. So just a little heads up there. I don't want you to get confused. So let's take a look at set 5 out of those in-class questions from your packet. And I'm going to give you a second to take a look at this and answer those 2 questions. Okay. So for number 1, if I remember my saying, never let monkeys eat bananas, then I know never eat bananas. Those are the granular ones. Let monkeys, those are going to be your agranular ones. So lymphocytes, remember that's going to include natural killer cells, T lymphocytes, and B lymphocytes within that category. Number 2, which of the following controls the production of white blood cells? Well, it's not aldosterone. That's got to do with blood pressure. Leukopoietin, I just made that word up. Erythropoietin, that specifically is red blood cell production. Erythro means red. So interleukins or colony stimulating factors would have been the right answer for number 2. And that concludes objective 5.